Okay, we're going to give everybody a little bit of slack here on this, so let's just get started, and hopefully we'll get everybody back. Everybody will be able to get back in. I assume those who can't get back in will let us know, Sarah, and we can do something to help them out.
Right. I'll take note of that for the states if there's any discrepancy in the report that I send out.
Okay, terrific. I appreciate that. I'm going to give you control, Rebecca.
Okay, our next speaker is Dr. Rebecca Curry. Rebecca is currently a senior toxicologist in the Regulatory Affairs Group at Arxada. She was formerly a toxicologist in the Product Stewardship and Regulatory Affairs Group at Eastman Chemical Company. And she was an assistant professor in the biology department at Roanoke College and a research associate at Virginia Tech. Rebecca earned her PhD from Virginia Tech, investigating ecosystem stressors from active and abandoned coal mine sites and industrial effluents. Rebecca is also certified as a senior ecologist by the Ecological Society of America, and Rebecca will be talking to us about hazards of working with pesticides. Welcome, Rebecca.
Thank you, Grady. I know it's been a long day for everybody, and some of you already have seen this presentation, so there'll be some things I'll try to go through a little bit more quickly and get the slides to advance. And Terry and I overlapped a little bit as well. Whoops. There we go. As usual, I always start out with this slide. I think it kind of emphasizes what a lot of individuals have covered today, and specifically with the need to make sure that you're following everything that's on the label, that you're actually reading the label. I'm sure that most of you are very familiar with reading labels and SDSs, so it's just a reminder of the importance of that. Obviously, EPA started their Read the Label First campaign in order to encourage that. But as an environmental toxicologist, ecotoxicologist, and as someone who, it's my job to generate a lot of these data that go on the SDSs, as Terry had indicated, as hazard data, and also someone who participates in the risk assessments and developing the classification signal words and the pictograms that go on a lot of these documents. To me, it's critical that you're paying attention to these and that you understand the information that you're looking at and the effort and time, I think, that went into generating a lot of these data to make sure that you're working safely and you understand the hazards of these materials. Ultimately, on the right side of the screen, you see the exploding chest pictogram. It's one of my favorites because it's very dramatic. But pictograms are designed for you to very quickly be able to look at an SDS and determine what types of hazards there are. So again, that you're working safely. Obviously, if you see this one, you should have an understanding of what the hazards are. In this case, it signifies that this could be a carcinogen, could be a sensitizer, it could cause reproductive toxicity, target organ toxicity, could cause mutagenicity with damaging the genetic material within cells, and it can contribute to aspiration toxicity. So very important to make sure that you're aware of them and you're paying attention to the materials that you're working with. It's a quick agenda. Whoops, my apologies. My mouse is a little quick today. Quick agenda review here. So we're just going to cover pesticides in general. I know Terry covered some of this information. We're going to focus mainly on discussing risk as it relates to human health. We'll talk about some of the hazards or the hazard data, the ecotoxicity and environmental fate and human health data that's generated, that's driving the information on the SDSs, that's contributing to the risk assessment in the identification of proper PPE. EPA, requires a lot of data in order to make a very definitive decision about risk and what types of uses and applications and PPE requirements go on these labels for these pesticides. Or you can think of it, I can call Terry if I know that she has something that's going through a re-registration process, and I can say, hey, Terry, we just ran a DAFNID study and I got an LC50 value for you of 1.5 ppm. milligrams per liter. And then that may be great, and we can send that study to the EPA, but it's a lot of other things that the EPA needs besides just that one endpoint to drive the information that goes on the SDSs, and again, to do the risk assessment to determine if the product is safe is based upon the use that it's designed for. We'll talk about exposure. Briefly, we'll touch on human risk assessment, and we'll cover some of the hazards and symptoms of pesticide exposure, and we'll end with talking about some of the wood preservatives. A quick definition, you've probably already seen this today, but any substance or a mixture of substances that's intended to prevent, destroy, repel, or mitigate any pest, very common. Pesticides are designed mostly to kill things. But the EPA definition can be a little bit broader than that. So we also consider a substance or a mixture of substances intended for use as a plant regulator. This could be plant growth regulation. It could be used, they could be used as defoliants or desiccants. There's a lot of plant growth inhibitors that are used by nurseries. We also use them in crops. They contribute, or they cause the plant to be shorter and stockier. So if you're using it on wheat or barley or oats, you end up with plants that are shorter in stature, have a thicker stalk, and you don't lose as much of the crop when you have bad weather. Nitrogen stabilizers also fall under this definition of pesticides. They hinder nitrification or ammonia volatilization. So there's other things just besides pesticides, or what we think of as typical pesticides, that fall under this definition. But realistically, for our purpose, these are going to be anything that are meant to control pests, right? The ICID ending, Latin, refers to kill things. So again, they're designed specifically in most cases to have a job functioning as weed control, like a herbicide, or they could be insecticides, repellents, insect repellents, animal repellents, the antimicrobials, the biocides, they all are included under our definition of pesticides. Again, pests are anything that's going to be harmful to us. It can cause different types of illness. It affects our crops. It can destroy foods, either in the field or in storage. They can affect our living conditions as well. So there's a variety of organisms that we perceive to be pests. There are different types of pesticides. This is at a very high level. There's the broad spectrum pesticides. These will be the organophosphates, carbamates, pyrethroids. We already heard Ryan talk about the neonicotinoid insecticides. They kill a variety of organisms. They're very effective, but the problem is that they indiscriminately can be harmful. So they kill everything. So if you're spraying along the roadway to get rid of weeds, well, you're going to get rid of good weeds, bad weeds, all kinds of vegetation. So they don't really they're not targeted towards a specific organism. They could be problematic in the environment. As you can imagine, if you're using a pyrethroid for mosquito control, I usually give the example, it happened in 2020 up in Idaho, but this happens frequently. While spraying for mosquitoes, if you're not careful, in this particular case up in Cass County, it was the typical time that the monarch butterflies migrate through that region, and they decided it was important at that time to spray, and they did. And of course, it killed all the monarch butterflies that were migrating through there. So hundreds and hundreds of butterflies, all over the county were killed. So they can be very detrimental if they're not applied properly, and there's not a lot of planning around using them. And we also have narrow spectrum or selective pesticides that target specific organisms, and examples would be the rodenticides, miticides, fungicides. And these are commonly used around homes and things. So you don't want to be spraying something on your yard if you're trying to control mosses or a fungus on some of your fruit trees or some of your vegetation, and end up killing all of your landscaping. So, it's important to make sure that you're selecting proper products for proper uses. They can be organic. There are several organic pesticides. Bacillus is a bacterium. It produces a toxic protein. It can be used against nuisance insects. If we introduce this bacteria, this toxic protein that the bacteria produces will kill those insects. Kaolin clay was approved back in 1998. It has kind of a smothering effect, so it's considered an organic pesticide because you can use it in there's a lot of variety of uses for it. And then neem oil is another example. This is obtainable at your local hardware store or nursery. A lot of people like to use it because, again, it's organic when they're wanting prevent pests from attacking their vegetables and tomatoes and cucumbers and things like that. But typically we're familiar with the synthetic pesticides such as the organophosphates. Common one is glyphosate, or what they use to make Roundup. And then organochlorine such as DDT is another example of our synthetic pesticide. One of the most unique things, and what makes pesticides so important for controlling pests, is that they are designed to have a very specific mode of action. They target specific sites within organisms. This could be a protein or an enzyme that's inhibited or blocked. For example, the organophosphates bind to acetylcholinesterase, and that controls nerve signal. So if you bind up that enzyme, the nerve will continue to fire, and you'll get an insect that will twitch and have uncontrolled movements. And obviously, it ultimately leads to their death. And another good example are the larvicides, which affect insects by acting as an insect growth regulator. So they mimic juvenile hormones. The insects never reach maturity, and so they don't reproduce. And so obviously you're controlling populations that way. So it's very effective against certain types of insects. Terry already talked about active ingredients. So the active ingredient has got to be listed on the label. It's the biologically or chemically it biologically or chemically acts against the target pest or pests. So it's kind of what's doing the job. And again, it's going to be labeled by its percentage on the label. Since we're going to be talking about risks, one of the things that EPA does consider when looking at active ingredients is whether or not there are metabolites or breakdown products that form. Some pesticides, when they're used in the environment, they'll actually metabolize into these byproducts that can be more toxic than the parent material. So that's one thing that contributes to difficulty with some risk assessments. For example, the biocide DBNPA is very effective in cooling tower applications. It really helps to control bioslimes, but it degrades, or it has byproducts that are more toxic than the parent material. So working with that material in a registration is very difficult, because you have to consider those byproducts or those breakdown products and what happens to them, especially in a situation like a cooling tower. It's that water from that tower, does it get treated? Where does it go? So it's a consideration as well. Another term when we're looking at pesticides is the other ingredients. This would be the inert ingredients. If they're proprietary, sometimes you won't see them listed on the label, or they'll just say other. And then, of course, as Terry mentioned, the end-use product is it's the final formulated product on the label, when it doesn't state that it can be used to manufacture or formulate anything else. So it's what you have to work with. Again, Terry had covered this a little bit, so I won't go into detail here, but all pesticides that are distributed or sold in the US, they must be registered by the EPA, and that falls under FIFRA. And the goal here is that when these pesticides are used according to the specifications, very specific, when they're used according to the specifications, they will not generally cause unreasonable adverse effects. And that's, as an ecotoxicologist, what I have to focus on. And EPA defines these unreasonable adverse effects to mean any unreasonable risk to man or the environment. And the EPA will also consider economic, social, and environmental costs and benefits of the use of this pesticide. And that seems a little bit odd, but when we get to the risk assessment paradigm, it'll make a little bit more sense. And it also, an unreasonable adverse effect can mean a human dietary risk from residues that result from the use of a pesticide in or on any food, inconsistent with the standard under Section 408. So we don't want all these pesticides ending up on different types of foods, because obviously that could be problematic as well. So the overall goal of collecting all this data, which will go on the labels and the SDSs to drive PPE requirements and the signal words and hazard statements, also gets used in order to determine risk. And risk is the probability of harm or an adverse effect if exposed to a hazard. So risk is the hazard based upon the toxicity data that we developed times the exposure. If you have high risk, no, I mean, sorry, if you have high hazard, if you have a high hazard, high toxicity, you have a pesticide that causes a lot of toxicity to fish, birds, but you don't have any potential for exposure. So you've done all this lab testing and you have all this data, but then ultimately there's no exposure potential, you have no risk. And alternatively, if you have minimal hazard, very minimal toxicity, and high exposure, the risk is low. So the EPA has to take this information and they have to make a very determined decision regarding risk. And it's become even more stringent and conservative since TSCA specifically was updated in 2016. So this is what the EPA risk paradigm looks like. On the left side, we've got the overall risk assessment process, and we've talked a little bit about this. So the hazard identification, this is the toxicity data that's generated through standard toxicity tests. We'll use those data, we will, and the EPA will, to develop a dose response assessment. So you're going to take a lot of information from a variety of different species and different study designs, and we're going to ultimately come up with an exposure assessment that says at this concentration, at this dose level, we see toxicity below this, it's likely safe for humans in the environment. So we have to take a lot of data and kind of condense it down into a usable number. And we'll also look at the exposure assessment, how long, where, how much, when you're looking at the environment, will people on non-target and target organisms be exposed. All this will be used to develop the risk assessment. From that, an EPA has to make some decisions, right? They're going to look at, is it a high risk, low risk? Are there control options? Are there legal considerations? And this is where the economic and social benefits and factors come in. Is it the only material we have that we can use in this heat transfer system? Okay, maybe it's persistent in the environment, it has high toxicity, but we don't have anything to replace it with. It could be a hydraulic fluid. A lot of times they're very toxic. If they get spilled, it could be a big problem for the environment, but yet we don't really have anything to replace them. So that could be a consideration when you're looking at economic and social factors. From all this, EPA will make their risk management decisions about applications and uses and things like that, and how we can move forward with registering these materials. So we have two categories when we look at risk assessments. We have the one for the environment, the ecological one, and one for human health. But the basic information for a risk assessment comes down to how much of a chemical is present in the environment, how much contact occurs between a person or an ecological receptor. So this would be the environment, target, non-target species, and the pesticide, and what is the inherent toxicity of the active ingredient. So that's just the basic information that EPA is expecting. And this information comes from the registrant, either during a new product registration or the re-registration of an existing product, which Terry had talked about previously. I won't go through this in great detail. Again, like Terry had covered a lot of this in really good detail. My job at Arxada is to step in when we have the official data call in. So at this point, when EPA looked at the existing toxicity data, this could be data from 20 years ago. Anything that they have, any new data that we have, they'll look at fate and physiochemical data. They'll look at the manufacturing process, where, how many people will be exposed, how is the material handled, is it coming in rail cars and totes, is it being formulated on site, how is that going to happen, are you pumping it out of vessels, how do you clean these things? They're going to look at the application rate, frequency, timing, method of application, what types of crops, what pests are you wanting to treat for, how will this material be stored and disposed of. And when they have all that information and they start looking at any gaps, then we will get an official data call in that says, okay, well, we feel that have enough chronic data, you don't have any marine data, you don't have, so we'll get a list of studies that need to be either waived if we feel as a company that we have enough data to support that, or studies that have to be placed and monitored and ultimately get sent to the EPA. And that's kind of my job, to make sure that the studies get placed and run properly and the data ends up with the EPA. All this testing has to be conducted following appropriate guidelines. Some of them are from European guidelines. A lot of them will incorporate OECD guidelines in addition to EPA. They're usually very similar, so we'll run them so we can use these studies in a lot of different geographies. They'll follow global laboratory practices. They result in a final report that gets submitted as a package to the agency, and then EPA will make an assessment of whether they think that it's an acceptable study. I just threw this in there because I think it's interesting from innovation of a new molecule, and we don't have a lot of new pesticide molecules, but on occasion when there's a new molecule, but going from innovation to market can take a decade, and it can cost millions of dollars. And even a standard or more typical data call-in for ongoing registrations can cost close to $1 million. I mean, it's not uncommon for me to have to place studies and spend $300,000, $400,000, $500,000 to get these studies conducted for the EPA. The toxicity data that's required for the data call-in, either whether it's for human health or the environmental aspects of it, will be determined by the characteristics and potential exposure of the pesticide. So they do consider data requirements based upon where the pesticide is going to be used and what the exposure potential might be. So if you're wanting to get a pesticide registered for use in Maryland, and you're treating tomatoes for some type of a pest, and there's the potential that, and actually this is a true story, if there's the potential for runoff to go, say, into the Chesapeake Bay, well, then of course the EPA is going to make sure that you have some type of marine data. So they're going to want to know, what type of aquatic data you have, how much of it's freshwater, how much of it's marine, and they'll ask for more additional data based upon a use that has been identified that might be problematic when you don't have data to support it. And testing a lot of times is done on both the active ingredient and the formulated product. So we'll talk a little bit about the testing, so you kind of understand what we're talking about when we talk about the endpoints that you can see that are on the SDS. Where do these come from? When we talk about acute toxicity, this is just short-term exposure. The endpoints are going to be generally reported as lethality, which is mortality, immobility, or impairment. From an acute standpoint, all of these are obviously detrimental to organisms. Studies can be 24 to 96 hours. There are inhalation type studies that can be shorter, from 3 to 4 hours. A lot of times these studies are just a single dose. If it's aquatic, you put it in, mix your solutions up, and you throw your organisms in there at different concentrations. If we're looking at a rat study or mice or things like that, rabbits, you'll have different doses that they can be exposed to in a variety of different ways. But if there's a potential for this pesticide to have toxicity, or they think there's an exposure issue with it, then we may have to run these studies with multiple doses administered over time. So these would be repeat dose studies. And if they're aquatic, then we may conduct them in a flow-through system rather than just a static beaker, or we may have to renew after so many hours, or remove those organisms and put them in new solution. Because what we're trying to be as environmentally realistic as we can. We generate endpoints that are time-based, and they show an adverse effect of 50%. So if you see something that says there's an LC50 value, then it was lethal. It was determined to be lethal to 50% of the test organisms. In the old days, they would do a XY graph, and you could interpolate where the LC50 value would be. Now we use software programs to do that, but they're fairly straightforward. If we're looking at an effect like immobility or an impairment, then that's an EC50 value. And when we're looking at dosing mammals, we'll consider it to be an LD50 value. Chronic studies are longer term. They're reported as sublethal effects. So we're not looking to cause mortality in chronic studies, but we're looking at those sublethal effects where we're just starting to see impairment, and it can be considered something like inhibition of reproduction, which is termed fecundity, or it could be an inhibition of growth. These endpoints are determined by the statistical analysis to the control organism. So when we're looking at acute values, we're doing this in this interpolation of time versus concentration, where we see this 50% mortality. And then we're looking at chronic testing. We're looking at these sublethal effects, but we're comparing back to control organisms. We report these as a no observed effect, adverse effect level. You'll sometimes see it referred to as a no observed effect concentration. But when you're looking at an NOAEL or an NOEC, that's a concentration that's not statistically different than the control organism's response. So for ecotoxicology and environmental fate studies, when we're talking about hazard, what types of studies are we running? Acute or chronic? We're looking at toxicity to organisms. If we're looking at aquatic toxicity to fish, invertebrates, and algae, this is considered an ecotox, a 3-pack, right? Because you got algae, which is your primary producers. You've got your invertebrates, which eat your algae, and then you have fish that can consume the invertebrates. So you're looking at 3 trophic levels, so it's called the 3-pack. All these are tested as either freshwater or marine species, and then we have aquatic plants like lemna or duckweed. And then for terrestrial toxicity, we've got all kinds of plants. If you're looking at agricultural crops, I mean, there's studies that will have 10, 15, 20 different types of crops that vary from tomatoes to ryegrass, and a variety of bird studies. They could be acute or chronic, different species. We do songbirds, we can do quail and duck. It's just a very long list of organisms. And then bees, we can look at larval bees, we can look at adult bees, we can look at residual studies and pollen and nectar. There's earthworm studies, different arthropod studies. So they're very complex in some cases, and becoming more interesting as we continue to develop new types of studies with different types of organisms. Environmental data are used in the environmental risk assessment to determine what's called an environmental effect concentration. So we'll develop a concentration that below which we don't expect to have any effects in the environment, and above which we have concerns. We have to take into account environmental fate. So biodegradation, hydrolysis, what happens when this pesticide gets in the soil or sediments? Does it absorb? What happens to it when it gets into an activated sludge system? I mean, you don't want pesticides running off into a wastewater treatment plant and then having trouble in the reactors or having any issue with the sludge. We're interested in bioconcentration. If something ends up in a stream, is it stay in the sediments and then ultimately move up the food chain, and we ultimately see it in fish? So is it bioavailable, and does it bioaccumulate and then become bioconcentrated up the food chain? And then we're interested in endocrine effects, which is a very big deal in Europe right now and becoming more of a concern in the US, although the EPA was really good about establishing their endocrine testing program many, many years ago for pesticides. So the EPA will also evaluate the potential for the active ingredient to be biologically degraded, to persist in the environment, bioaccumulate again in the food web, and if there's any potential for endocrine disruption. So all this has to go into the risk assessment. For human health, we've got what's called the 6-pack for human health. This is the minimal amount of data that's required by the EPA. So we've got oral, dermal, and inhalation toxicity. We look at both eye and skin irritation, and then skin sensitization in a number of different mammals. These studies provide the initial data that are used for classification and the precautionary labeling, hazards, and the PPE requirements on the SDSs and the labels. But for human health, there are also subchronic studies. These are conducted as repeated exposures over a limited period of time, so they can be, it doesn't sound like a limited period of time. I mean, you're looking at 28 days or 90 days in a repeated dose, and they can be in multiple species. And then we can look at multiple routes of exposure. They can be exposed orally, dermally, or by inhalation. And they include an evaluation of body and organ weights, and there's a necropsy to examine organ and tissue health. So these are becoming more complex, looking at organ systems as a whole to look at for any subtle impairment to, again, make this evaluation of risk and how these pesticides should and can be used. There's also what's called the genotox battery. So we're looking at an Ames test, which looks at bacteria, in vitro mammalian mutagenicity. These are looking at generally a human cell line, I believe, in these, and chromosome aberration would also use some type of a mammalian cell line. So these are kind of benchtop studies to look at what's actually going on in the genetic material of these organisms or these human and mammalian cell lines. And then there's chronic testing. When you get to human health with the mammalian studies and you start looking at these long-term effects, and then you're looking for developmental impairment and damage or changes to reproductive organs or fecundity, they become very involved. And they can be multi-generational, looking at development and reproduction. So you're actually looking at effects to fetuses when exposed to when you expose pregnant animals. So you can imagine these, you're using these bred animals that you know are carrying fetuses, and you're exposing them, and then first after a certain period of time, you're allowing these pups to be born, and then depending upon the species, and then you're looking at them for any developmental impairment. There are neurotoxicity tests, mutagenicity tests looking specifically in cells' genetic material, and then you've got the endocrine assays, which are considered a higher tier testing, and then at the top you've got cancer studies, which we don't see a lot of cancer studies because they take a very long period of time. They're very difficult to interpret, and they can cost upwards of, they could be millions of dollars to run these studies. But they were going from these more simple studies like the Ecotox 3-pack and then the 6-pack for the human health into these more complex studies as we try to determine the risks. So anytime we see any type of impairment or effect, and then we have to go on to more additional testing. This is just quickly the dose response relationship. When you're looking at all these endpoints, he had mentioned that the LD50 was a lethal dose to 50% of the test organisms, and you can see down here on the x-axis that it's showing the LD50. It is about 22.5 maybe. Again, you could hand draw these if you want to, but this is kind of nice. It's got a nice S-shaped curve here, but it also shows the NOAEL and the LOAEL. So you can see those chronic studies, when we look at that no effect level, we're not looking to determine mortality, we're just looking to determine an effect. And so generally, these chronic studies are conducted at lower concentrations below where you would expect to see an LC50 or an LD50. I included the PNEC, which is used for the environmental risk assessment. I don't have it on the graph, but it's a predicted no effect concentration. It aligns with the DNL kind of, which is on here. The DNL is a derived no effect level above which humans should not be exposed. It's a very complicated calculation to determine the DNL. But if you look at this DNL, you're down at about maybe 3. When you have a NOEL here, you're no observed effect level at about 10. So you can see how much more conservative that DNL value is. Generally, that DNL value is going to drive the risk assessment. So it's a super conservative value. It's got assessment and safety factors added to it to have that very conservative approach to make sure that at that DNL below that, you can feel very confident that there's going to be no issues and no potential exposure problem for humans. But above it, it can be problematic. So we've talked a little bit about hazard. We'll talk about exposure. We're getting close to the end. We'll try to wrap this up because I know, again, it's been a long day for you guys. So risk from exposure is affected by individual overall health. All right. So a healthy person can be exposed, and the risk may not be as significant as if someone who is more susceptible that may have an underlying health condition, could be pregnant, could have allergies, could be very young, could be very old, and gender can have a difference. So all these things can affect the exposure potential or the risk from the exposure. So emphasis is given in the risk assessment to protect susceptible populations, and these would be children and pregnant women. So there's always this extra safety assessment that's applied in order to make sure that we're protecting those populations. So we also have to consider duration. How long is the exposure? The frequency, is it single or repeated? Obviously, longer duration or more frequency can increase the risk. We need to know the concentration, the amount that's you're being exposed to. This can be in parts per million or parts per billion. and the dose. When the dose is the amount that actually enters the body and is usually adjusted for body weight. So you'll see it reported as milligrams per kilogram body weight per day. Just as a side note, not all doses of a toxin or a substance are harmful. You can overdose an aspirin. Supersaturated environments of oxygen can be toxic. Interestingly, apple seeds contain cyanide. You don't really worry about eating an apple, so in small amounts, apple seeds are perfectly fine. I don't think you would want to eat, 100 apples a day and consume the seeds. Could be problematic, but it's just this idea that lethality is determined by the dose, right? Dose makes the poison. It's kind of our take on a saying. BPA is a good example. Realistically, there's no health risks to consumers because it's too low. Your exposure to BPA is too low. But again, some of these things are driven by other issues. On the right side of this figure, also, water can be toxic in high enough concentrations. Caffeine, I think it's 118 coffees at 240 mils can be toxic to some people and cause some type of an effect, other people not. And then alcohol, you may be able to consume 13 shots of, alcohol, sit at the bar with your buddies, but somebody else may only be able to consume 8 before they start to see alcohol poisoning. So, the dose really does make the poison, and that's something that we have to manage when we're looking at pesticides and registrations and trying to determine risk. We have to consider the routes of exposure. So are we looking at inhalation through the lungs? Are we looking at a dermal exposure, which can be contact? So you get that contact dermatitis, you get a rash, or you can see absorption through the skin. Ingestion obviously is problematic. You don't want to be consuming any of these materials. When you're looking at getting pesticides or active ingredient or any type of chemical in your eyes, you can have damage specifically to your eye if it's corrosive, or you can have materials be absorbed into the body through your eye as well. The route of exposure will be dictated by the type and the use of pesticide. So, what's design use, how you're handling it. For applicators, the dermal exposure is most common. It's the one that obviously it happens the most, but it can be dealt with either using proper PPE to avoid having that exposure or immediately going into some type of a safety shower, just removing that material from your skin surface as quickly as possible. Inhalation is less common, but obviously it's going to pose more of a serious risk. You can jump in the safety shower, but once you've inhaled these materials, aspirating it can be very difficult. So when we talk about human health risk, and since we're more concerned with making sure that everyone's working safely, not worrying at this moment about the environment, but they're very similar. So it's easier just to go with this one. But the 4-step process, we look at hazard identification, which we've talked about. It's the toxicity of the active ingredient and/or formulated product. So you have your toxicity data. You develop a dose response assessment. So you're looking at this numerical relationship between exposures and effect, and you end up with this estimated, in humans, you end up with an estimated equal dose in humans. So we want to be able to take all this data that we have and make sure that we have adapted fish, bird, and rat data, that we're able to use that and come up with a numerical value that's going to be protective to all humans. We look at the exposure assessment, examining the frequency, timing, and level of contact. Then we come up with the risk assessment, if you remember the paradigm, and always throw in some applications of the appropriate safety factors. So we're going to have safety factors adjusted in there to be conservative. And the ultimate goal is risk management. Okay, so this ends up in your hazard warnings on the labels, your PPE requirement, and then again we can have restrictions on volumes, restrictions on uses, and applications. For an example, a human health risk assessment may determine that there's a high risk for inhalation toxicity during commercial use of handheld spray equipment. All right, so the EPA may restrict this end use of the product and only allow brush-on applications, or they may require that a full face respirator has to be worn. At that point, it's a decision of whether or not that's an application that you want to continue to support in the registration. If it's for commercial use, then you may worry that your consumers are not going to want to switch to a brush-on application, or potentially your product doesn't work as well as in using it on a brush, or you may be okay with requiring the full face respirator on your label and your SDS. All right, so we'll go over some of the hazards, try to wrap this up for you guys here. We talked about dermal exposure. So we can have these contact adverse effects, right, in dermal exposure. This can result in a rash or a burn to the skin. And again, you can absorb material through your skin layer, and it may result in dermal absorption into deeper tissues, right? So you can have just this surface contact, you know, you've got a little poison ivy and it's itchy or scratchy, or you can ultimately, you know, have a problem deeper in your tissues because you've absorbed this material. You can have systemic adverse effects. So you can get a localized exposure that spreads to the other parts of your body. So you know, you touched up against something, you had an allergic reaction to it, and you get the blisters and things like that, and all of a sudden you're itchy everywhere, right? So you can have a systemic reaction. Or even, it doesn't have to be an allergy, it can just be a systemic effect. And then there's also allergic adverse effects, which can be systemic, which I just mentioned, and can be contact. So rashes, hives, anaphylaxis. You can become sensitized only after one exposure. So it's very important to minimize your exposure. And these are not dose dependent. So when we talk about allergic reactions, you don't have a worse allergic reaction because you were exposed to more of it. It really doesn't, it's not dose dependent like that. Some of the symptoms of pesticide exposure. So this may occur immediately or hours after exposure. And to keep in mind, again, this is just making sure you're aware. Obviously, most of you are probably very well aware of these hazards, but just to kind of go over this. So mild poisoning, this can be a little bit more difficult. You may go home with a headache, you know, and just think, gosh, I worked really hard today, or it was just really stressful, or, you know, you just have a lot going on at work or at home. Dizziness, loss of appetite, thirst, nausea. I mean, you know, maybe you're just not feeling all that well, but you really need to be keeping in mind if you have these, and especially if they're persistent symptoms, that, you know, you may have been exposed. If you take these mild symptoms and you have any of these what are considered moderate signs of poisoning, a little bit of blurred vision, you've got it coughing, excessive salivation, I mean, you start vomiting, that's always a concern. You know, obviously you need to be thinking about this. You know, have I had the potential to be exposed to a pesticide that I may have been working with today or yesterday? And then by the time you get to the severe poisoning list, you realistically most likely know that you've been exposed to a pesticide. And, you know, all of these should be taken very seriously. But by the time you get to the third column, you really are probably in some type of distress and should be seeking medical attention immediately. Obviously, if you have chemical burns on the skin, and a lot of these are things that should be taken care of immediately, you know, if you've been exposed, to avoid any potential prolonged problems. So if we just kind of have a brief discussion on the hazards of wood preservatives, obviously wood preservatives are pesticides. The toxicity is going to be determined by the specific active ingredient, so you should know what active ingredient you're working with, regardless of the product name. The route of exposure is going to be dependent on contact with a formulated product or the article. Most risk is from personal exposure during the application process. And exposure can take place while you're handling and mixing chemicals, when you're entering pressure treatment cylinders, working around spray or dip operations, handling freshly treated wood, cleaning, servicing equipment, or disposing of waste. So any of these activities, you have the potential for exposure. So you need to be thinking about that and reading the labels and the safety data sheets, so you're understanding, you know, everything that you need to do to be safe. You know, wear the recommended PPE, follow good housekeeping measures and good hygiene. You know, you're not drinking or eating in the working areas, you're washing your hands, you're removing your gloves when you're done, changing clothes when necessary, and things like that. So you could be exposed realistically at any point in time, even when you're not directly working with the active ingredient. I'm just going to wrap up here with a couple of examples of things to think about when you're working with these materials. If we're talking about CCA, which is, if we look at the active ingredient list, you're going to see chromic acid, arsenic acid, copper oxide. Hopefully, if you're working with this, you're reading the label, you've read the SDS. You may think that, well, it's chromic acid, arsenic acid, copper oxide. How bad is that really? You know, trivalent chromium is an essential trace element in humans. Chromium as a pure metal has no adverse effects. So when you think about chromium, we don't think about it being that hazardous. But hexavalent compounds, or hexavalent chromium, can be toxic. It can cause different types of issues, dermatitis, allergic reactions. So hexavalent chromium can be problematic, but chromic acid is corrosive and is considered a carcinogen. So this would be included on the SDS for you. When you think about copper, we usually think, well, copper's not really a problem. You know, we use copper pipes for water. You can suck on a penny all day, it doesn't really bother you. But copper can be very toxic to aquatic organisms. It's used frequently to control things like algal blooms. So, you know, it may not be a problematic for human health when you're working with it, but it's not something that we want released into the environment because it can be very toxic to aquatic organisms. So it can, from an environmental risk assessment, you know, copper's problematic. But when you're looking at CCA, one of the biggest concerns is the arsenic compound, okay, for both the human health and the environment. So the DNL for arsenic is about 12 micrograms of arsenic per meter cube. This would be for inhalation. That's 12 ppb, parts per billion, so that's really low. And then mortality has been seen at 600 micrograms per kilogram per day. Again, it's a very, very low value. So although we've got 3 different active ingredients or components that are making up the CCA, it's important to kind of have an understanding of all of them and how they contribute in different ways to toxicity to either the environment or to you. If we looked at arsenic SDS information, this is what you'd see. And again, you may look at this and say, well, I'm not really sure what an acute aquatic toxicity category 1 is. These are things that I work with our regulators and our regulatory affairs people to put on our SDSs and things. It means something to me. It may not mean a lot to you, but anytime the numbers are low and they're at like a 1, it's not good. But we do have the warning statements on here to help with that. So if you can consider it to be acutely and chronically toxic to aquatic organisms, and it does have oral acute toxicity and toxicity by inhalation. And then, you know, you can look at the statements to see it's very toxic to aquatic organisms, can cause long-term adverse effects. You know, so there's all this information that you can use. And the pictograms, I think, you know, kind of tell the story too. You've got the dead fish, dead tree. And then when you see, you know, skull and crossbones, that should, you know, really be a warning sign for you without having to see the word, you know, danger or poison. If you know you haven't taken the time to read the SDS. This was mentioned earlier that the proper PPE is outlined in Section 8 of the SDS. In this, I know this got cut off here, but it should say manufacturing facilities may have PPE requirements that are more stringent, right? And so you always want to make sure that you're following the most stringent PPE requirements, whether they're on the SDS or not. And then observe the pictograms and warning statements. Again, I use the exploding chest. There's 8 that are mandatory. The 9th one is the dead fish, dead tree. I don't know why, as an environmental person, it's not a mandatory one, but you don't always see it, but you will see the warning statements about toxicity to aquatic organisms, and you'll see these categories up above as well. And I think this is the last one. I won't go through details. I know I've been talking for a while, but again, you know, the SDSs and labels are available. They're outlined in sections that provide, you know, really all the information you need to be safe when you're working with these pesticides. You know, we're encouraged to read them. If there's any questions, you have a whole list of folks at Arxada. They're more than happy to answer questions for you. And I think that is the last one.
I do not have any questions for you, Rebecca.
Their heads are all down at this point, and they're thinking, oh my God, is it time to go home? Of course, they're in California, right? It's a little earlier in the day, but still, long day, I'm sure.
A long day. Okay.
Sarah, do you have a poll question?
I do. I'm going to relaunch that poll just in case the states have a question about that, so let me do that now.
Hopefully it'll work.
Hopefully it won't get stuck on the screen. Okay. All right, we're at 80. Oh, wow, everybody's doing great. 92% at 100% true. 100% voted, 100% true. Excellent.
Fantastic.
Okay, that's the way it's supposed to work. Thank you,
Very
Grady.
good. Okay. Thank you, Rebecca, for that presentation. Our next speaker and last speaker is Dr. David Jones. David is a treating division manager for timber products inspection. For that position in April 2021, he served as the director of project services, BMI, for 6 years. He also was an associate professor and extension specialist, forest products at Mississippi State University. David received his PhD in forestry and wood science from the University of Georgia, his MSF in forestry from Stephen F. Austin State University, and his BS in forest resources at Clemson University. He is a member of several trade professional organizations, including the Society of Wood Science and Technology, AWPA, International Association of Wood Anatomists, and the North American Wholesale Lumber Association. David will be talking to us about a successful in-plant quality control program tonight. Thank you, David, for joining us.
You're welcome. Glad to be here, and as y'all can already tell from what Grady said, I'm pretty nerdy overall, but a lot of you will already know me from overseeing some of your plants. Those of you that don't, you're aware of the program that we do. And so what we're going to do is kind of go over what the elements of successful in-plant quality control program is. And overall, it's broken into three areas that we're looking at primarily. The first part is sampling, and we're going to talk about what the purpose is, what are the standards, what methods do we use, and what equipment we need for that. I'm more of a simple, put things straightforward kind of person. Sampling purposes, one's to demonstrate the product meets the specification. We all want to make sure that we get a product out that meets the minimum, at least the minimum requirements, so that it performs out in the field like we want it to. The second part of sampling is ultimately to control the process. The more control we've got over the treating process, the more efficient that we can be, which leads into the next component, which is to manage resources. Even though some folks might not want to hear it, one of the things that this does is it allows you to hone in on your chemical usage so that you can meet the requirements with also out overusing chemical. But the biggest thing, and why most people do it, it's required under the rules and regulations set forth by AWPA. Many of the chemical manufacturers require sampling as proof that their product's being used efficiently and correctly. And ultimately, it's just a good idea to perform periodic sampling as needed. Now, the standards for sampling are set under AWPA's rules. First is the U1, which is the user specification for treated wood, and that details what the treating levels are that are required. T1 are the rules that are associated with processing and the actual treatment standard. Thou shalt do this to meet this specification. Those samples are then analyzed using what's known as the A standard, which are the analysis methods that are associated with the specific type of chemical that's used. And ultimately, there's two things that it can be broken down into when you're looking at standards. And that is what we consider the M standard, or the M standards, which M2, which is for industrial use materials. You've got M3, which is the standard for other products, and then M25, where I misspelled standard, so I'm obviously not as nerdy as I thought I was, for quality control and inspection of preservative treated products in residential and commercial use. So what are the basic equipment that we need in a facility to be able to perform sampling appropriately? Well, the first is to have what's there in the upper left-hand corner there, directly under the word sampling. And that is when I went to forestry school, we always called this an increment boring bit. In the treating industry, we generally just refer to it as a boring bit, because we're not doing trees, we're actually sampling lumber. You have to have one of those bits. The rules state the preferred diameter for that bit, and it needs to be at least long enough for you to get the sample depth within the product that's described in the standard. Now, the old school way of using that boring bit, you either used a T-handle, which clamps on the opposite end where the threads are, which I will tell you is a very labor-intensive process that makes you have really big forearms. After that, they had the bracing bit handle that worked, but now most people use an electric drill, often a battery-powered electric drill, because the technology's come up, which then leads you to the middle upper picture, which is the adapter that goes into an electric drill that allows you to turn that increment boring bit and then use the spoon or the retriever to get the increment core back out of that hollow sampling boring bit. Ultimately, you take those, place them on the cutting block, which is upper right-hand corner of the slide that you see there. Depending on your facility, you may require, in this case, this is a tray that does 20 cores, but I do know that some facilities use 30 core trays because they take 30 cores as opposed to 20. Under the standards, 20 is the minimum number that you can take to determine whether or not a charge has been properly treated. You'll see that there are lines that are cut into that block. Those lines are there to help you determine whether you've met penetration, and then to help cut the assay zone of the cores off at the proper depth. And you'll see that in the bottom left-hand picture, where we've got that tray filled with cores and a utility knife that inspector was going to utilize to cut off the assay zone. Now, in that process, once the cores are taken, you're going to need an indicator, one for heartwood to determine if there's heartwood in the particular wood that you've treated, and then some sort of indicator for the preservative that you're utilizing in your treatment. And so in all of this, the equipment that's shown here is the very, very minimum that you can have in order to acquire samples at your facility. When sampling, it's very important to follow a couple of simple rules. The first is obviously you should not sample the top and bottom boards when you're working with 2-inch material, primarily because those boards are likely to get a little more chemical than the boards towards the center of the pack on the outside. And so what you want to do is skip the top and bottom boards and then set you up a pattern, a systematic pattern. You don't want your QC people to go, well, I'm going to take the third board this time, and then I'm going to skip 3 more, and then I'm going to skip 5. You want to have a prescribed methodology set out to assure that you're getting a random sample from your charges to assure that they're treated properly. So it's best to set up a sampling pattern, whatever that choice may be for you, and to sample properly. Now, one thing to mention at this point, the right place to sample on a board is in the center of the board. So, you want to be equal distance from both ends of the board where you sample. Because if you sample towards the end of the board, obviously, you're going to get a different result than if you sample towards the middle of the board. So it's very important to set up this pattern and have it ingrained in your personnel's mind as what the proper way is to do it. Doing it completely random is not an appropriate way, because human nature is to do the easiest thing a lot of times. And so that randomness often leads to people not sampling effectively. Once the samples are taken, they're placed in the core. Again, it's important for you to determine what the appropriate depth is to take the cores. Then you generally apply the Heartwood indicator, allow it time to react so that you know what part of the sample is heartwood and what part of the sample is sapwood. And then you determine penetration using whatever the chemical that is required to do the reaction to determine how much penetration you are able to get in that particular charge. Go back, because I got a little happy with my clicking there. So again, proving that I'm not as nerdy as I thought I was, because I just messed up a presentation. But as we go on, once you've taken those samples, you have to move on to analysis. And there are ultimately 3 things that we look at when we're doing analysis to the samples. That is the sample prep step, and then the actual analysis, and then in part of that is doing the appropriate calibration and verifications. So for sample preparation, everybody doesn't necessarily use the same methodologies, and so and that's often because of the equipment that they have on hand. Ultimately, minimally, you have to have 2 of the 3 that's shown on this, but the others are wisely used. So for grinding, we do have some facilities, know of some facilities that use coffee grinders to grind their samples down. We prefer that not to be the case, only because coffee grinders tend to give an inconsistent consistency to the cores once they're ground. Same goes with a flour mill. Ultimately, the best method to grind the samples is to use a Wiley mill. It doesn't take up very much space, and I have to admit it is kind of a expensive investment initially, but it's very rare for a Wiley mill to go out, and if it does go out, it's easily fixed and serviced. The only thing that I generally see go out are the sieve tubes that go into the Wiley mill itself. Now, you also have to have a method to dry the cores. And while you can use a microwave to dry cores, if you're unlucky like I am, every time I've ever tried to dry a piece of wood in a microwave oven, I always catch it on fire. So I just stay away from using microwaves. You can use a kitchen oven or a countertop oven to dry cores. Again, you have to make sure that you set the appropriate temperature to assure that you're reaching the zero moisture content for the particular wood samples. The best is to use a laboratory style convection oven because they generally are able to hold temperature more consistently, and there are also not any hot spots or cool spots within those ovens as a general practice. Now, as another part of that, in your method, you need to have at least a thermometer to be able to measure your oven temperature to assure that it's reaching and holding the appropriate temperature. You need sample pans. And then for certain X-ray machines and for certain chemically treated wood products, you need to have a press with the appropriate torque wrench to assure that you're pressing to a consistent torque there. And so it's very important. If you should know, both from your chemical manufacturer and from your third party, whether or not you need the addition of the press. Now there are 4 basic machines that are out there, and 2 of which are actually the same manufacturer, but it's a changeover. So you see the Spectra and a Soma. They have not made any new models since the Phoenix 2. You've got Rigaku, which is a common model that we see out there. A lot of mills have the old Oxford machines that seem to be pretty bulletproof and last a long time. Oxford was bought by Hitachi, and so there are no new Oxford machines, but the Hitachi machines run very, very similar to the old Oxford. So if you have to replace an Oxford, then it's good to follow up and go with a Hitachi, since the similarity between the two. Now, each one of those, the manufacturer has got recommendations of how to handle samples, whether you need to press samples, whether you don't need to press samples, and how much sample material is required. I recommend contacting your equipment manufacturer to determine the appropriate steps that are needed. You can also reach out to your service lab, and they can discuss what the appropriate methodologies for your equipment are. With XRF, there's kind of a dirty secret to XRF, and it's not really dirty and it's not really a secret. So I'm not sure why I said it that way. But XRF ultimately is only doing an estimation of the amount of copper or other heavy metals in the sample. And because it is only doing an estimation of that, you have to create a calibration utilizing known standards, so material that has been treated to a known retention level. And then you adjust and make the calibration based on those samples. And then that will then give you a good calibration for the material. And then ultimately, once you've created the calibration, then you have to do some sort of verification where you utilize a standard chemical analysis method to verify that what you got out of your calibration matches up. Again, it is very wise, whoever your third party agency is, that you perform periodic comparisons with them to assure that your calibration and their calibration fall within a reasonable relationship, so that you're not under treating material and you're not over treating material based on the XRFs. So basically that information is run a couple of different ways. Obviously using an agency, an independent third party. There are 3 third parties out there right now that do preservative treated wood. They are Bodie, which is in the Pacific Northwest. There is SPIB, Southern Pine Inspection Bureau, that owns Western Wood out west, but they are one entity. And then there's the company that I work for, which is Timber Products Inspection. All 3 of us are certified under the ALSC blanket to be able to do third-party inspections. 2 of us perform ESR inspections under the ICCES protocols. All of those protocols follow the AWPA standards. And again, the third parties evaluate both M22, M22, and M23 to assure that facilities are followed up on. Now, for poles and cross arms, many of those fall into the NRECA wood quality control program that the co-ops go into. And so, again, that's another level of oversight. This particular, not for commodity type products, but more towards the poles and cross arm programs. So inspection agencies are accredited. Generally, again, we carry both accreditations, both from ALSC and from IAS, which indicates that we're ISO certified to be able to do the inspections. There are 2 types of program inspections. Kind of already mentioned this earlier. It's ALSC, which is the quality checkmark program, and then the ESR program, where you have an ESR number, and then it basically follows a very similar pattern to the ALSC because, again, it follows the AWPA standards. You can do independent and lot inspections. It does not mean you have to necessarily be in a program. Under the ALSC program, it is allowable to do lot inspections, but you cannot tag material with the quality check mark unless it has been inspected if you're doing lot inspection type stuff. Most inspection agencies offer laboratory services. One that we discussed earlier, Bodie actually uses another laboratory to do their program. But again, the other 2 agencies have their own internal laboratories to perform the analysis of the wood products. So what are the standards that the third parties use? The biggest one is the M22 standard, and this was just updated last year. And what we look at with the M22 is first we measure the ability of the facility to consistently treat, and that's done by calculating a lower confidence limit. So that lower confidence limit sets a minimum associated with how out of control a system can be based on the retention levels that are measured. It's calculated on the most recent 20 charges, and it's calculated for all preservative components. So if you're treating with MCA, MCAC, CAC, or other chemicals, each one of the components of those chemical makeups has to meet a minimum LCL requirement. As part of the M22, there's also the evaluation of average percent core conformance. Now, where I live in Georgia, obviously the prevalent species is southern yellow pine. And as many of you know, southern yellow pine tends to take preservative treatments much more easily than the refractory species found on the West Coast. And so for y'all on the West Coast, sometimes this APCC plays a role in that. It is calculated again on the last 20 charges. 80% of your material that is sampled has to have passed penetration. So if we take 20 cores, at least only 4 out of 20 can fail. If you had 5 out of 20 fail, that throws you out of that 80% conforming, and a charge would fail. But if you fall out of that with your last 20 charges, let's say that you go to 79% of your material is passing, that is below the allowable limit for APCC, and that would trigger the facility to be placed into increased, which leads to the next component. As part of the inspection status, it's calculated on LCL and APCC, and basically the M22 has 4 levels, and that is routine, which means everything is chugging along at the plant at a regular rate. The LCL is above the appropriate lower limit. The APCC is above where it needs to be. If you fall below the LCL or the APCC, you would move into an increased status. And so long as every charge that you treat after you go into the increased status passes, you will stay in increased status until your LCL returns to above the minimum or your APCC returns to above the minimum. If you do have a charge that fails while you're in increased, you will move into warning as your status. And again, you will stay in warning until your LCL comes up, and then you would move back into increased or your APCC, depending on which one triggered the increase. If you get those back up, then you'll move back into increased. You can't jump right back into routine. If you then have a charge, if you're in warning and have a charge fail, then based on the rules, then you become disqualified. And in order to requalify, you have to have 20 charges that the calculated LCL is above the minimum required by the regulation. And so it can take a long time, depending on the size of your mill, to get back into qualification. Now, if you remember back, there's an allowance for lot inspections. So it is possible to still tag material, but that material must be inspected and pass with your inspection agency to continue tagging it. And that means that only material that has been inspected and passed may be tagged until you requalify. Here's a good example of a LCL graph. You'll see here the blue dots are the individual charges. The red triangles with the green line, that is your LCL. The red line is the LCL minimum. What you'll notice here on this graph, this has changed. That red line is at the minimum retention. The changes that happened mid-year last year required the LCL to go up from the minimum retention up minimum retention plus 5%. So now for 0.15 material, the LCL minimum is 0.16. So it's very important to understand that if you're treating to the bare minimum of 1.5, you can never get your LCL to 0.16. So it's very important to understand the kind of the calculation behind that. This is kind of a quick and easy schematic that we've got drawn up talking about the different statuses. Again, If you go into increased, but all your charges conform on the next inspection, but your LCL or your APC doesn't come up, then you stay in increased. And again, it shows you the overall progression, moving from routine to disqualification, and also moving back up from increased back to routine and warning back into increased. So this is a helpful thing that we give all of our clients to be familiar with. It is written out very clearly in the AWPA standards. Overall, we've got kind of a cheat sheet, because part of the regulation that we have to follow is M23. And M23, again, you can see denoted by the 21, it changed last year. And so it's much, what I would say is much meaner. The old standard allowed you to accumulate points, and so long as you never went over that threshold, you never changed in status. You can have one issue in the process group, and you can see on the left-hand side there the process group items, and not really change in status. But if you have one in the product group, then you can move immediately from routine status into warning, which triggers a faster turnaround for the inspection agency to come back in. And it also means that you move towards disqualification. So even if everything is perfect in M22, if you move in status, you can move in status to disqualification very quickly in M23. That would mean 2 inspections where there was an issue with the product group. One thing to note is most facilities are doing the quality control appropriately in the facilities, so that this rarely shows up, but occasionally things happen, like treaters not paying attention to the charge numbers, and they become illegible, so you can't track material, or somebody gets a little gung-ho about putting tags on material, and material gets tagged before it has been proven to be conforming. Or more often than not, what happens is we run into people who have tagged material, then it comes back that it failed for retention, and they don't remove the tags immediately from that material that needs to be retreated, which causes a problem with the product group there. Again, like M22, we've got a movement in from M22 routine status to routine follow-up status, to warning status, to disqualification. The big thing is there are a couple of big jumps pretty quick, where you can go from routine status to warning status, and then to disqualification very quickly. So it's very, very important to understand M23 has got a little more teeth to it than it used to, and therefore it's very important to stay on top of it. So overall, what have we talked about? We need to make sure that you're following sampling methodologies that are outlined in the AWPA standards. And one of the things is to make sure that you have the proper equipment to do the testing that you need to do. Everybody's required to do sampling of the material and verification of it. This also includes making sure that you have the proper calibrations and verifications done for XRF machines that you have in plant, and for some other chemicals for HPLC, if you do have those. You need to make sure that you've got an effective internal quality control system in place, and that it is done appropriately and consistency. One of the biggest issues we run into is consistency, particularly with the way the job market is right now. And turnover rate is that you get somebody trained in QC and then they leave, and you've got 2 weeks where you don't have anybody trained up to do QC, and so things fall through the cracks. The way I like to look at it is you should always have a bus idea in your mind, and what I mean by that is if your QC person got hit by a bus on the way to work, would anybody else in the facility be able to perform that job? And so it's always good to have some sort of redundancy in your system, that you do have somebody as a backup, so that you don't get behind, because the last thing you want to happen, and the last thing I want to happen, is for you to have a bad inspection because you were unable to perform the QC roles that you needed to. The important thing is to control what you can control, and that is ultimately the analysis and the sampling that you perform within your facility. And if there are any questions, I'm glad to handle them. I made sure it was short enough so that if there were any, if not, a lot of y'all know how to reach out to me, and obviously if you're using another agency, you can reach out to them to get more information about sampling and inspections and process controls within your facility.
David, I don't have any questions.
Okay,
Thank you.
you're welcome.
Thank you, David. We appreciate you joining us and giving us that good information. Sarah, any more poll questions, or are we finished?
We are complete. We have no more.
Fantastic. I'd like to thank everyone for attending this year's virtual West Coast Plant Operators Seminar. In particular, I'd like to thank Belinda and Sarah, who have done a wonderful job over the past months, putting all this together, getting state approvals, and working with issues today all day long and making it seem like it's easy. You can expect a certificate to be emailed to you shortly after the seminar. And on behalf of Arxada and our presenters, who we thank very much, thank you for joining us today, and have a great rest of your day, what little is on the East Coast.
Give me one
Any
second,
other-
Greg.
Okay, you got more?
I wanted to thank you very much for being our moderator. You've been doing this for us for a couple years now, and we really, really appreciate the efforts that you make, especially because today you even had to do a presentation. So we really appreciate your time all day long and the effort that you made. Thanks also to Brian Delbrook. I know he's not on the call anymore, but for his keynote address, and thanks to Eric for his opening comments, and actually to all the speakers. We really, really appreciate your time.
Thank you, Belinda. Okay, if there are no questions, we're going to sign off.
All right,
Thank you,
everybody have
everybody.
a great year, and we'll see you next year.
Yep, we'll see you next year. I will submit all of your rosters to your states, so there's nothing else for you to do.